So my name is Saul. I'm an undergraduate in the Hariharan lab. I'm going to tell you today about regeneration in Drosophila. So regeneration is the regrowth of lost or damaged tissue. Many organisms have the ability to regenerate damaged tissue. A couple examples of this include newts and salamanders, which are able to regenerate entire limbs once they're severed. Lizards, which are able to attach their tails to avoid predators and regenerate the lost tissue. And the human liver, which can regenerate the entire organ from as little as 25% of the initial tissue. Now, unfortunately for us, the process of regeneration is poorly understood. We hope by studying organisms that have the ability to regenerate will give us a better understanding of how it works. Now, also unfortunately for us, many of these organisms that have the ability to regenerate are not very good for lab work. But luckily, Drosophila melanogaster, commonly known as the fruit fly, is a very common lab organism and also has tissue that is able to regenerate. They're good for lab work because they're easy to breed and maintain, they reproduce quickly, and they have lots of genetic tools that are available to work with. So, a little bit of background about Drosophila. They go through a series of life stages, from embryo to larva to pupa and then to the adult fly. Now, the stage that's important to us is this larval stage. During this larval stage, they have these small sacs of tissue called imaginal discs. Imaginal discs eventually turn into adult structures when the larva matures into an adult fly. So here we have two wing imaginal discs that will eventually turn into the adult wing when the larva matures. Additionally, different parts of the imaginal disc turn into different parts of the adult structure. So the area marked in green here, the wing pouch, eventually turns into the adult wing blade. Now, it's been shown over half a century ago that wing discs are capable of regenerating tissue. So as you can see here, when we physically cut a wing disc, over time, the damaged tissue will regenerate and reform a complete wing disc. Additionally, we're able to use immunofluorescence to look at gene expression within the disc. So as you can see here marked in green, this is a gene called puckered, which is known to be involved in regeneration. And as you can see, in the tissue that's regenerating, we have puckered expression, whereas in the rest of the disc it doesn't seem to appear very much. So we hope that understanding why certain genes are on and other genes are off in regenerating tissue will give us a better understanding of why some tissues, such as the human liver, can regenerate, while other tissues, such as the human heart, cannot. Now, the previous methods for studying regeneration in Drosophila were very difficult. They involved dissecting wing discs out of larvae, surgically cutting them, and then in order to keep them alive so that we could study regeneration, you had to implant that wing disc into the abdomen of an adult fly and later attempt to recover it. As you can imagine, this process was very laborious. It involved a lot of dissections of these tiny little tissues and transplantations into adult flies. This just tended to kill a lot of the adult flies because they're sticking foreign tissue into them. And it made it very difficult to study regeneration in Drosophila. So, what we've done is we've made a new genetic system that allows us to study regeneration without the need for all these dissections and transplantations. So the way it works is we're able to cause ablation within a specific tissue in the wing disc, this area marked in green, which is the wing pouch. And this is all in response to a short heat shock, which essentially equates to taking the flies and raising the temperature that they're raised in from about room temperature to 100 degrees Fahrenheit for about half an hour at any point during development. And what happens is this heat shock causes the activation of these ablation genes, which cause cell death. So what we can do is we can kill the cells within the wing pouch and then allow the wing pouch a chance to regenerate. And if you'll recall, the wing pouch turns into the adult wing when the larva matures into an adult fly. So in order to see how well this tissue is regenerated, all we have to do is wait a little bit for the flies to mature and then see how well wings regenerate. And by looking at the size of the wing, we can see if we have any regeneration, partial or full. This means that we can cause damage and study the regeneration of that tissue without having to do any dissections, any transplantations, which makes the whole process a lot easier and a lot quicker. Now, additionally, we're interested in the genes that are involved in regeneration. So we made it so our system is also able to knock down the activity of the gene. We're able to do this by inhibiting the expression of the gene via an RNAi, which stands for RNA interference. Essentially, this works by preventing the RNA that should be coding for a protein from doing that, which then means that the gene that made that RNA has no activity. So from this, when we damage tissue and knock down the activity of the gene, we would expect to see one of two things. If we inhibited a gene that's not involved in regeneration, we would expect full regeneration within the wing pouch and adult wings that look fully regenerated, indicating to us that our gene is not involved in regeneration. Alternatively, if we knock down a gene that turns out to be involved in regeneration, we would expect that the wing pouch wouldn't be able to regenerate because it no longer has the supporting gene, and we would see adult wings that indicate that we have little to no regeneration, telling us that our gene must be involved in regeneration in some way. So, the majority of my work this summer has been around building and testing this system. First, we want to show that we're able to cause damage in cells in response to a heat shock. So, as you can see on the left, when we don't heat shock, we see no expression of these ablation genes and consequently no cell death. Alternatively, as you can see on the right, when we heat shock the larvae, you can see in the wing discs that we get expression of these ablation genes and cell death. This tells us that our system is sufficient to cause damage within the wing disc and that we're able to control when this damage is caused, so we can then study regeneration. Now, we've also noticed age-related decline in regeneration in flies. So the y-axis is how many wings, percentage total, and the x-axis is how well those wings regenerated. As we would expect, when we don't cause any heat shock, we see all wings are fully formed. And alternatively, when we cause a heat shock early in development, we see most of the wings are fully formed, with a few that have slight imperfections. This is more or less the same in early to mid-development, but once we start getting to mid-development, we see more wings have imperfections, and some have even partial to little to none regeneration. Now, this is even more apparent when we cause a heat shock late in development. This trend is consistent with previous trends identified in studies of Drosophila regeneration, and this indicates to us that our system is working properly and that we can therefore study regeneration with it. Now, additionally, we want to be able to knock down the activity of a gene within our system. So here we use wingless, which is a gene known to be involved in regeneration in Drosophila, and that has related genes that are known to be involved in regeneration in many other organisms, especially mammals. So the disc on the left shows the typical expression pattern of wingless when it's not knocked down. Of particular interest is this area marked in white, which is the wing pouch. We get a nice theta pattern of wingless expression within the wing pouch, which is what you would expect to see during regeneration. Now, when we cause knockdown using a wingless RNAi, as shown on this disc on the right, as you can see in the wing pouch, there's no wingless expression. We lose this theta pattern. This is telling us that our system is sufficient to knock down the activity of this gene in the tissue that we specify, so in this case, the wing pouch. So, while we're able to independently cause the expression of these ablation genes and the knockdown of the gene of interest, the important part is that we're able to do this in the same tissue. So, as you can see marked in green here, this is where we're able to cause damage and get regeneration. The area marked in red is where we're able to knock down the activity of the gene using the RNAi. And as you can see from the overlay here, the two overlap really nicely. This means that we're able to cause damage and then knock down the activity of a gene in the regenerated tissue. We can then study how well that tissue can regenerate without that gene. Now, additionally, our system allows us to knock down genes in different patterns within the wing disc. So here we can knock down once more in the red, but in this case in half of the wing disc, and then the other half without the gene knockdown. Now this means we have an internal control within our system. So half of the disc where we don't knock down the gene should be able to regenerate completely normally, and the other half where we do knock down our gene, we can study the regeneration in it. And by directly comparing how well this half of the disc can regenerate to this half of the disc, we can see what that gene's activity must be in regeneration. So, what we want to do with the system is to run a screen that allows us to identify genes that are involved in regeneration. So, as you remember, it's easy to induce damage taking only a 30-minute heat shock at any point during development that we choose. It's easy to assess how well the tissue is able to regenerate, since all we have to do is look at adult wings. This means that we can look at lots and lots of genes roles in a really short amount of time. And luckily for us, we have these libraries of RNAis that are available for our use. So we can systematically go through the entire Drosophila genome and knock down genes one at a time and see each gene's role in regeneration. And then by simply looking at how well the tissue can regenerate without that gene, we're able to say some genes will be involved and other genes will not. So, in conclusion, what I've shown with the system so far is that we're able to kill cells in a specific tissue in response to a short heat shock. We're able to knock down the function of the gene within a specific tissue. And these together mean that we're able to knock down the function of the gene in this tissue that we kill and is then regenerating. and then we can assess that gene's role in regeneration, with hopes that by finding new genes that are involved in regeneration, we'll get a better understanding of the process itself. So, in conclusion, I'd like to thank Dr. Robin Harris, Dr. Iswar Hariharan, and the whole Hariharan lab for support and guidance over the summer, and the Surf Rose Hills program and Nathan and Leah for making this all possible. So, any questions?